And today I'm going to be going over magnesium regulates cytotoxic functions of NK and CD8 cells in chronic Epstein-Barr virus infection through NKG2D. Written by Chain Delaland et al. Uh, so essentially, I'm going to give a little bit of background on XMEN disease itself. It's best defined just by its acronym. Essentially, it's X-linked inheritance, exemplified by this pedigree here. It being more likely to be expressed in males as is recessive, and women are likely to be carriers. And if both of them contribute a mutated X link, of course, it can be even expressed in women. And then magnesium defect actually is a major component of the immunodeficiency. I'll discuss later on this MAGT1 mutation affecting magnesium influx into the cell. And then we have Epstein-Barr virus itself. So essentially, this magnesium is needed for intercellular activation of immune cells such as NK and T cells. In their absence, Epstein-Barr virus and other viruses are more likely to infect and even lead to pathogenesis in these infected patients. We have the Epstein-Barr virus also contributing to the latter portion here of neoplasms. Epstein-Barr virus is linked to several lymphomas, and many of these patients will develop these in later age due to this inability to protect against the virus and others. And this is actually characterized through life. I showed this picture here, XMEN disease, a couple of their features. So they'll have a lot of ear infections, especially at a young age, and pharyngitis, molluscum. Susceptibility to other viruses as well, as of course the T cells being a major component of intercellular defense, intercellular pathogen defense, and then several patients will have splenomegaly, these two being heavily diagnostic, that enlarged spleen and that decreased CD4 versus CD8 ratio. Okay, and then I'll be going over the prevalence of this disorder essentially, as it was mainly discovered in 2014-ish. They don't know how predominant it is in the population. The number I saw in 2016 was about 15 diagnoses, but that was like mid-year. How prevalent it is now that evaluating for it is better, I cannot tell you. It's still unknown, really. Background on the study itself. They were trying to answer the question how the magnesium is related to the pathology in XMEN disease. What's the effect of it on Epstein-Barr virus? Why is it so essential, essentially? And purpose is to exemplify its function in XMEN disease. And the hypothesis was, after this, was that supplementation of magnesium in MAGT1 deficient patients can restore expression of this receptor here that I'll discuss in the next slide. So some of the key parts of this study to understand is the MAGT1 gene itself. It's a family of receptors in charge of magnesium influx, as pictured here. And it's basically expressed on all human tissues, but there's several other forms of receptors that get magnesium into the cell, so it's not essential for too many, but it's especially important for several cells of the immune system, like T and NK cells, as it's part of the activation, and magnesium is part of the activation of these cells for them to take their effector functions. And it's now actually classified as a tumor suppressor gene due to this fact. Of course, T cells in and of themselves can suppress tumors as well, and then of course the virus also being linked to it. So there's two mechanisms of neoplasm in this disorder. Then there's the receptor itself. This is just one of the receptors that can be found on T cells, and even NK cells will express them more later than not. Basically, it helps them identify infected cells and target them for killing. Several cells have this ligand here that'll let them know that they're infected, and it'll communicate to T and NK cells that they are a target for cell death. The main proponent of this study as Epstein-Barr virus is heavily infected B cells, and B cells being the only portion of the immune system that these, or the adaptive immune system that these patients can exhibit strongly, it being another huge factor. but they'll express this to T cells for them to be targeted for death, especially in Epstein-Barr virus. DAP10 is an important part of this complex in the receptor too. It's essentially for this interaction. It wasn't discussed too heavily, but I figured it should be worth discussing, as it also has a part of this interaction. Interferon gamma also wasn't really talked about, but I figured it was worth mentioning, as NK cells heavily express them, and that communicates to macrophages and T cells that there's a problem in the body for them to target, and helps signal where that is, of course. It's a cytokine, and T cells will actually express it later on in the adaptive response as well. Gaps in research that this was trying to fill. They knew that XMEN disease is associated heavily with Epstein-Barr virus. They knew the normal functions of NK and CD8 cells and how they help protect the body. And they understood that blood-borne extracellular magnesium was normal in these patients, but it was lower in the cell itself. Hence, they figured that these were related, and they wanted to understand why this magnesium contributes to the pathology of XMEN. Okay, and then a couple of the aims in the experiment. They want to understand why it's not controlled in the disease, of the Epstein-Barr virus specifically, and how magnesium has to do with that. They want to understand the ion movement, again, to understand the magnesium's part in this stimulation of NK and CD8 cells. And they wanted to see the effect of magnesium supplementation in vitro and in vivo on the control of the Epstein-Barr virus, measured by lysis of infected cells. So this is the table I'll be discussing mostly, as I figured it was the major groundbreaking thing they discovered. Figure 3 discusses it more in vitro. A and B through C actually explain the expressivity being deficient of XMEN of these protein, NKG2D. I believe most of these were beta cells, which was of high importance because, of course, the beta cells that being infected with the Epstein-Barr virus. And it measured the receptors frequency outside the cell. There were some other ones they wanted to rule out as well, and they saw that XMEN patients, the controls, and this is just the background. Just really just the isotype in the gray here is just serve as a marker. And then so the only one they saw a difference was the NKG2D subtype being altered, as the blue here representing the XMEN patient expressing it much less frequently, as shown by the fluorescence in this flow cytometry, and then the normal patients having normal amounts and much higher, as signified to the right of the frequency, higher fluorescence, and then the rest being relatively similar in both controls and XMEN. And then B shows that again another huge difference between the expression in XMEN versus normal, and even they can also compare it to other diseases. This XLBP is another disorder that's X-linked called Duncan syndrome, but it's separated here by this component of the disorder. Obviously, a difference in phenotype. And then this is just a normal patient with a chronic active Epstein-Barr virus. They still can express the protein to several orders of magnitude more than the XMEN patient, but as you can see, all of these are segregated by their expression. And then this shows gel electrophoresis of the protein and being the normal wild type receptor, and this is an altered form found in XMEN, showing that normal patients in these two columns here having not having this receptor, altered form of the receptor, while a huge expression of the normal subtype, and then XMEN will have a deficiency here and expressing mostly this defective form. And they'll even have a complete absence, or well, not complete, but near absence of DAP10 as opposed to the normal patient. And this is in both cytolytic T cells and NK cells. Then we can look at D and E, which deal with before MAG supplementation, the percent of lysis and their ability for the mutants to target this Epstein-Barr virus specifically, shown later in E. But in general, the ability of this cell to target cells for lysis is much diminished in patients of XMEN versus normal. And then even what's shown here is that comparing the percent of lysis with this ligand here that's mostly associated with Epstein-Barr virus lysis. It's when it's uh, uh, actually the major component of the lysis, hence associated with Epstein-Barr virus destruction. That's a major component of what the patients are trying to do. They're still trying to fight off the Epstein-Barr virus, but it's not near to normal levels of what should be done to keep the cell in control. Then C is more of a flow cytometry, but it compares, this is MF4, which is a marker for magnesium on fluorescence, and then this is FR, which is a marker for calcium being important for the activation of CD8 cells as well. Basically, it's mag flux that leads to K flux from the sarcoplasmic reticulum via exchange. And then we'll have the ratio rise as magnesium supplementation rises nearly, it's very much associated almost linearly, as you can see here. So this ratio becomes near normal. Magnesium rises, and especially relative to calcium, which is normal in the cell. And then we have that in CD8 versus NK cells, translating to this biomarker, or this marked NKG2D receptor rising in expression, as exemplified here as a increase of this hill of cells expressing much more of this marked NKG receptor, even in T cells, but also in NK cells, much more increase of expression of the receptor. G shows after MAG supplementation, showed by this positive and negative in a normal control patient. It won't really make a difference. Adding magnesium to a patient with the defective allele will show an actual increase of expression of the normal receptor, here shown on this channel column here, and then even diminished of the altered form. And even DAP10 will come back after MAG supplementation, but this is a major component for the NK cell, as I'll discuss further. Okay, so some conclusions they were able to draw. Epstein-Barr virus is kept in check by NK and CD8, interferon gamma, MAG-T1, and NKG2D expression. Supplementing MAG restores NK function, but it does not actually aid T cell function, they found, or even the MAG calcium flux. This is important because it actually it shows that MAG cannot affect one arm, but basically the aspects that control Epstein-Barr, but it can help the NK arm. So NKG2 expression helps control Epstein-Barr virus levels in vitro and in vivo. And the summary that I found was basically this supplementing XMEN with magnesium increases the lysis of Epstein-Barr infected cells by upregulating this receptor here, as magnesium can now enter the cell, helping even through this division transporter. But it's more specifically on NK cells, not really aiding too much with T cells. Alright, so this is the current proposal of the mechanism. I actually took this from a separate article that I have cited later on. Essentially, both the NK cells and the T cell part of this helps control Epstein-Barr virus, which Epstein-Barr virus is linked to lymphomas. This is the two things that suppress it. MAG supplementation, shown here, will help increase the expression of NK, or the NK function of controlling Epstein-Barr, but doesn't really help the TCR. This is still being studied into how this affects the clinical effects, essentially. But it was interesting to know that this article does show that it can help the NK cells. Okay, so this is some other research that I helped get some of the figures from, and then this is a trial here that I found of importance as well. It's due in June of 2020. It's showing a large trial of patients getting supplementation with MAG and showing whether that translates to clinical. Not much is discussed yet, but knowing a patient of this disease, his physician contacted the trial runners, and it seems that it's actually not being significant in help aiding the prognosis as of yet. Likely showing again that this T cell arm is of utmost importance, but I'm definitely curious to see what they say in June. All right. Thank you for your time here and everything throughout the semester. I hope you have a great rest of your day, and thanks for everything.